Hi, my name is Sara, and this is my science fair project Bioengineering alter genetic mutations and create organs using stem cells. A lot of information was omitted to ensure my presentation is less than 10 minutes. All relevant information will be shared at the in-person fair. My project is designed to study how bioengineering can be used to alter genetic mutations and create vital organs, but my project extends to serve bioengineering's purpose to find the most affordable, comprehensive, ethical, and inclusive way to create these vital organs and modify genetics. The objective is to contribute to mitigating the organ donor crisis through biologically engineered organs. I hypothesize the combination of fresh, focus rotary adjustment, and 3D printing techniques derived from origami will become a leading method for bioengineering vital organs using stem cells due to its potential to address several challenges. I also hypothesize that CRISPR-Cas technology, accompanied with other genetic modification techniques like like effector nuclease, will allow for the alteration of genetic mutations and the treatment of genetic diseases. Tissue engineering and regenerative medicine integrates biology, engineering, and biochemistry to repair and replace defective organs. Bioengineers create functional tissues by combining scaffolds, cells, and physiologically active chemicals. Serum is used to create functional human trachea, skin grafts, arteries, cartilage, and bladders. These have all been successfully transplanted. Eukaryotic cells tend to cluster when introduced to each other and produce an extracellular matrix to support themselves. The idea is that when you get a cut and cells are rapidly performing mitosis, they're actually creating new skin. So we're thinking, can we do that on a large scale? So scientists can mimic this ECM with polypeptides obtained through 3D printing and manipulating amino acids by creating peptides. Upon fabrication, researchers place these scaffolds into laboratories with environments stimulating cell proliferation to promote tissue development. Stem cells and tissues can be salvaged, which is my idea, from surgeries to cultivate growth and specialize in other patients' organs using their own stem cells. So this was basically the idea where you take an extracellular matrix. This is not my idea, obviously, but you would put it through ionic and anionic determinants, decellularize it, then you'd have this matrix, and then you put in an environment that promotes cell proliferation, and then you recellularize it. With scarce biological materials imperative for tissue engineering and regenerative medicines, many scientists are resorting to bioprinting to mitigate the crisis. Bioprinting uses a layer by layer approach that involves building from a base of polymers, which eliminates the challenge of encouraging cell development and biologically fabricated scaffolds. Biological printers have two main approaches when producing scaffolds: acellular and cellular constructs. The acellular constructs serve as a supportive environment and they do not have the cells planted in them. And then cellular constructs already have them and basically are printed with the cells. So they serve as a template for cell attachment, which stimulates tissue creation. So CAD, which is found in this image, is just the computer generated. And here, pre-processing is just the computer thinking, what am I going to do? What parts of the coordinate plane am I going to align? And then they create the construct. So acellular structures have great precision and consistency and have been able to make muscles, liver, cartilage, bone, and skin. But despite their capacity, biomolecules loadings are frequently necessary to insert cells or molecules into the scaffold. Another possibility is to include cells and molecules directly into it, but this is very difficult to print. It's very difficult to replicate the texture and tenderness of human tissue, which increases the risk of the body identifying these organs as foreign objects and consequently rejecting them. So what about Fresh? Fresh is about 3D bioprinting techniques used to fabricate scaffolds made of soft biomaterials such as alginates. Alginates are extracted from brown algae and other marine organisms that are known for their gel-forming abilities and biocompatibility. The Fresh technique involves continuous extrusion of bioink through a nozzle to create a 3D structure. Bioink is composed of alginates in the cell and a supporting hydrogel, which is continuously cooled during the extrusion process to prevent premature cross-linking and gelation. Following the extrusion process, the supporting hydrogels actually dissolve using a solvent, leaving behind the actual 3D scaffold, which can be post-printing processes can enhance the mechanical process. But the issue is, in short, is just cell viability and long-term scaffold stability, which means it's just not structurally integral and it's not sustainable. So this is actually the process. So they actually print it in, they freeze and lithosize it to ensure that there's no cross-linking, and then now they will recover it and bring up the temperature, and now you get the culture cells, and then you repeat the process. So focus rotary adjustment was used to fabricate the first human ventricle model composed of helically aligned beating cardiac cells, which imitates the heart helical structure and enable it to distribute blood through the body effectively. Focus rotary adjustment provides fibers using a centrifugal force on liquid polymer, which is pushed through an opening and then allowed to evaporate using concentrated stream of air positioning the fibers through the process, through the vesicular collector and then through the stream. But what Harvard John A. Paulson School of Engineering students decided is that they positioned it at a 30-degree angle, which helically aligned it. So instead of going like this, now it's going like this. This is actually the structure of a heart. So the human induced pluripotent cells were inserted in the fibers, which led the ventricle to beat. And the students examined the ventricle's features and found that ventricles produced with helically aligned fibers beat those made with circumventionally aligned fibers in every single trial. So bioprinting would take 100 years to replicate this. It's just not viable anymore. The focus on design is very effective to produce complex collagen extracellular matrix in the heart, and induced pluripotent stem cells are feasible because they need their connection to capillaries, and bioprinting would just take too long. They need to have their natural way system, and focused photodiesterase can actually facilitate that. So this is actually the process, helically aligned structure. This is the formation, the patterning, and then this is the structure, and that's what they created. And this is actually what they created. It's very cool. I recommend watching their video. Genetic diseases and stem cell involvement. This is the second part. So hemopoietic stem cells actually treat genetic mutations. So genetic mutations are characterized by single gene mutations, alterations in chromosome structure, and mitochondrial genome anomalies. So environmental factors like toxins and infections, and psychological factors and nutrition and scarcities can actually impact this. So stem cells can provide a produce-specific cell within an organism and be used to rebuild the blood and immune system after a high-dose chemotherapy and a lot of myelogenicities and genetic abnormalities. So bioengineering has made tremendous advances in treating blood abnormalities, and actually it's now completely approved by the FDA and everything to treat hemophilia. So hematopoietic stem cells, which are cells that serve as the foundation for all adult stem cells, are critical to the success. So this was actually one trial before stem cell therapy and after stem cell therapy, which increased bone density exponentially. So how can we apply this? So CRISPR-Cas9 tailings are used for genetic modification in the treatment of hereditary diseases. So CRISPR is simple and efficient, but can sometimes cause unintended cuts, leading to mutations. Tailings are very specific and less likely to cause off-target effects and mutations, but are more difficult to design and construct. Scientists can use a combination of CRISPR and tailings for precise genetic modification. Modular recombination is also a process. So it uses a DNA repair pathway that is activated when DNA is broken. So after this cut, and it's treated by introducing a double-strand breakage, which is DSB, at a specific location at the genome when CRISPR-mediated genome editing. So by combining CRISPR's tailings and modular recombinations, scientists can achieve precision and accurate. So I would say modular recombination can then be like the safety relay that joins them if there's an unintended cut. And that's kind of what I've been talking about. You can read it. This is the benefits and cons and details that I've been speaking about. So how can we genetically alter the stem cells and make them perfect for the organs we want to make? So CRISPR-Cas and talons are used separately to edit these genomes, but combining them may allow for more precise and reliable. So I had this idea that we can actually combine these methods, and we can delete genes associated with disease or mutations, and inserting genes associated with a specific organ's function, we can create stem cells with the desired genetic makeup. So in 2019, a PNAS study reported successful use of CRISPR-Cas to edit stem cells for heart tissue creation. The edited stem cells differentiated into cardiac muscle cells and showed the potential of CRISPR-Cas. So once seeded into the scaffold, the cell cells differentiate into specific cell types depending on their environment and ultimately construct the harvested organ. But now that we can genetically modify them, we don't have like a 50-50 chance. We have a confirmed chance that it'll become a heart cell. This is a pig trial where they use different things like acellular scaffolds and some things I have not studied, which are like VEGF. Social issues and disparities. Racial minorities are faced with a stem cell disproportion. Ethnicity is a variable that affects the acceptance of stem cell donations, as individuals with similar ancestries have HLA proteins, which is what the body realizes, oh, is this mine? So if they see a lot of matching, they're like, yeah, I can accept this. So white Americans dominate the stem cell donor registry, which means other minorities and are less likely to receive biologically engineered organs. And although family members can donate, only 30% of patients discover a sufficient donor within their family. PhD student Jordan Wilson admits that the only way to alleviate this discrepancy is donations from the affected minority groups. Racism in healthcare has been documented to result in unequal processes of delivering, access, and receiving healthcare. Healthcare staff who are also racialized minorities experience racism in the workplace from both healthcare users and colleagues. Research on healthcare staff's racial attitudes and beliefs display that they exhibit negative stereotypes and implicit racial bias towards racialized minority healthcare users. The lack of stem cell donations consisting of minority groups contribute to stem cell shortage in certain ethnicities with matching HLA proteins. So this is a very basic rundown on it, and it's basically saying these shapes that don't match, they are rejected. So, in bioengineering, ethical issues arise with the application of induced pluripotent stem cells and human embryonic, and including the potential hazards they pose. So, bioethics within are far more debated because people don't know. Are we really okay with destroying an embryo just for genetic modification? And bioethics is very important because the Abrahamic religions forbid genetic alteration and cloning. Informed consent is crucial in discussing the ethical concerns of genetic engineering and bioengineering. So germline editing creates the most discourse as it's hereditary, and people down the line cannot consent because they're not born yet. So, a lot of germline therapy and funding has been consequently withdrawn from Canada. In the United States, it's not doing any better because, as you can see, the highly religious Americans are more likely to see gene editing for babies as taking medical technology too far. So, what's next? Combining the guiding characters of CRISPR, the precision and reliability of Talon's scissors, and the homologous recombination correcting capabilities, I believe that bioengineers can utilize them when constructing organs composed of genetically modified stem cells. So organ donor shortage can be attenuated through the combination of fresh, focused adjustment and bioprinting to produce organs, and modifies extracted cells by pluripotent and directed into specialized into cells found in the organs at hand through Talon's CRISPR and homologous recombination. In 2021, 7,220 Canadians required an organ donor transplant, with only 39.3% receiving a viable organ. The remaining 57.2% remain on the waitlist and 3.5% dying prior to receiving. Bioengineered organs with consideration of bioethics and perfected stem cells can increase the number of successful transplant and stem cells can be derived from the recipient's body, exponentially increasing the number of successful transplants. Are there enough recognizable HLA proteins? So this is just an outline in the database that I took my information from. Healthcare disparities, ethical conundrums, and limited accessibility can hinder the potency for a lot of these techniques. So we need to address social issues surrounding bioengineering, which can further the scope of it. To reduce racial bias, the healthcare workforce must be diversified, and we must include culture competency training, which I'll speak about more at the fair, to really dismantle structural racism. To mitigate ethical concerns in genetic engineering and bioengineering, it's essential to educate communities on the science behind genetic alteration technologies, their benefits and potential risks through public information campaigns, workshops, and even events in school. So, if I were to repeat this project, I would attempt to network with bioengineering students, such professionals, and discuss their outlook on my research. In addition, I will try to run clinical trials with willing professors and test my technique for bioengineering organs using a minimized scale and testing proliferation before attempting life-size models. Although I have attempted to contact various universities, including the University of Calgary, Mount Royal, Johns Hopkins, and John A. Paulson Harvard School of Engineering, which was a stretch, to participate in a survey and allow me to work alongside their professor, I did not have any prior experience in their research and laboratory experiment, and they were very reluctant to work with me and basically said no. The Calgary Youth Science Fair can provide me with the connections and tools for pursuing my project, and my goal is to have my research and proposals recognized at the fair so I can improve my project through clinical trials. I'm so excited to give you an extensive rundown at the fair. Have a great day, and thank you so much.